I would like to thank and welcome everyone to this session. This is a great pleasure to be in this new session, session number 67 from the International IVF Initiative. So for the videos and previous sessions of these meetings, you can go to the ivfmeeting.com, and please don't be shy about leaving a review on the videos. You can see every single video that's been recorded since the beginning. And again, today, I encourage you to ask questions in the Q&A section. Please do not use the chat function, use the Q&A section, because this is the best way for us to capture everything and make sure that we can address your question either by writing or live. And I would like also to thank Liesl Neeltemat, because she's the one really behind this nice program that you're going to have today. And she worked hard really to prepare everything, but unfortunately, she's so busy that she cannot be with us today. But we really thank her sincerely for that. So my name is Pierre Comizzoli, and it's my great honor to be a co-moderator with my dear colleague Gabriela Mastromonaco. And so without further ado, I would like to briefly introduce Gabriela. Gabriela is a scientist, of course, and she got her PhD in Guelph in Canada, and then she joined later, after multiple professional experiences, she joined the Toronto Zoo in 2007, where she managed the reproductive science department. And she recently took a very important position as the director of conservation science at the Toronto Zoo. And she has a very broad spectrum of expertise, from somatic cell nuclear transfer to endocrinology in multiple species. So thank you, Gabriela, for being my partners in crime today.
That's lovely. Thank you, Pierre. It's an honor to be here with you today. And of course, for everyone who's joined us, my co-host, no stranger to I3, is the much accomplished Pierre Comizzoli, and a pleasure to be here with him today. So Dr. Comizzoli began his career more than 25 years ago as a research veterinarian in wonderful places like South America and Africa, places we can all dream of going or being, and some of our participants have joined us from those locations. After completing his PhD in 2000 on in vitro fertilization in bovine and deer species, he moved on in 2002 to the Smithsonian Conservation Biology Institute in Washington, DC. He works on amazing and wonderful projects, new cryobanking projects on gametes and gonadal tissues from rare and endangered species, which also includes reproductive monitoring, artificial insemination in different species. We're talking about giant pandas, clouded leopards, Eld's deer, and many more. Since 2007, he's been leading a Smithsonian-wide initiative to improve the management and use of frozen biomaterial collections within the institution. So thank you so much, Pierre. We're going to have some fun today. It's time to move on to part 2 in our Therio genealogy series on animal reproductive biology, entitled Therio, Lessons from Animal Diversity. This time, we're going to take a look into different reproductive phenotypes, cloning, and transgenics from the leading names in animal conservation, reproduction, and genetics.
Exactly. So that's a very exciting session with 3 outstanding speakers. And without further ado, I would like to introduce the first speaker, Dr. Martha Gomez. So Martha is a veterinarian who trained in Colombia. But then after that, she completed a PhD in Sydney, Australia, joined the Louisiana State University for a postdoc to work on embryo production in ungulates and cat species, including also assisted reproduction and somatic cloning. She worked at the Audubon Nature Institute, as well as the San Diego Zoo. And now, actually, that's a beautiful kind of a next step in her career. Now she's working as an embryologist at the University of California, San Francisco, developing human embryonic stem cells from vitrified human embryos. So, Martha, the floor is yours.
Thank you, Pierre. It's a pleasure for me to be here today and being able to talk a little bit of previous work that we did at the Audubon Nature Institute in New Orleans. Basically, we established a program for testing the somatic cell nuclear transfer to produce wild felids, and being able to demonstrate that this tool can be used to preserve endangered species and probably in the future avoid extinction of some animals. Unfortunately, the center was closed in 2015, but I can tell you that those were the most rewarding 18 years of my career while I was there. I'm going to talk a little bit about why we ended up doing somatic cell nuclear transfer in wild felids. When I arrived to the Institute in 1999, Dr. L. Pope and Betsy Dresser had already established most of the technologies in assisted reproductive technology to produce wildcats by vitro fertilization, by artificial insemination, by culturing embryos. But at that moment, we were thinking, okay, there is another alternative that we could use, because since there are not too many wildcats, small felids specifically, that we can use and working at the zoos more in captivity, which technology we could use to produce animals. And we decided, okay, somatic cell nuclear transfer could be a nice tool. And by the way, then we decided it needs to be interspecies somatic cell nuclear transfer. It cannot be interspecies because we cannot collect oocytes from these wildcats. Then at the Institute, we have roughly, by then, 150 domestic cats that they were oocyte donors. And then we move on with the project. The project started in 2000, I think we started the project. At that moment, we didn't know which wildcat species we should use, because we could use any anyway, because we need just a simple cell. But we need to be sure by then that the domestic cat carry the pregnancy of those cats. In the 2000, Dr. L. Pope produced African wildcat that was born by interspecies embryo transfer, that was born after he transferred embryos in a domestic cat. Then in that sense, the project opened a path of using the domestic cat also as a recipient of these embryos. And we use also the African wildcat because it's the closest species to the domestic cat. As you can see here is the Felis libica, and actually the domestic cat, sorry for the redundancy, but it was domesticated from the African wildcat. Then we knew that the reprogramming of the nuclear somatic cell could be easier if we got closer in the phylogenetic. That was our first step to start the cloning project. Then simple, most of you maybe have heard done it everything, because it's a lot of people that have done cloning. Cloning is not as everybody expects in the terms of what do you need in a lab. In the lab, you need normal micromanipulated microinjectors that most of the IVF people use today to do ICSI and biopsy embryos. Also, you need to electrofuse incubator, but the main source that we need is a small tissue. We could use any tissue, but then we choose a skin to isolate fibroblasts from skin samples, because they are easy to collect. Any animal can die on the wild. You can collect it, even you can freeze a piece of tissue. Then you have, and there are strong cells to grow in culture. Then after that, you establish the monolayer of any of species that you want. We did a lot of research about which is the best cell cycle, how to synchronize these cells to have the optimum stage that we thought by then, and other researchers already demonstrated for nuclear transfer. Most of our work was done with in vivo mature oocytes. We were lucky enough to have a nice colony of cats to induce oocyte retrieval, and LPOP has already established this. That was easy for me. Okay, let's go to in vivo mature oocyte, better quality of oocyte for nuclear reprogramming. And the domestic cats are induced, hormonal induced with normal FSH, LH, and then we perform the oocyte retrieval. The method of oocyte retrieval, also developed by LPOP, was similar to the human IVF, where you have your forces, your trochers. One of the trochers contain your camera, the other contain your forces, and here are your Barrett's needle, where you connect the gas to inflate the abdomen. And through the abdominal, we puncture the wall of the abdominal wall and insert the follicular aspiration needle. After that, you have your ovary that we grasp it to avoid movement and collect in vivo mature oocytes. Okay, then you have your oocyte, you have your cells that you have already established your cell line. I'm going to pass a quick video of the method. There are several methods for enucleating the oocyte. I was using the most common method, that is where you have your oocyte here and you expose it to HESH to being able to see your metaphase and your polar body. With your enucleation pipette, you inject it and remove a little bit of that cytoplasm that goes out with your metaphase 2 and hopefully with your polar body. If your polar body doesn't go out, it's not, you shouldn't be too worried as long as you are sure that you are not fusing that polar body when you are electrofused. Then you confirm that you have removed or enucleate both of them, and then you inject your single cells. Another small project that we did with this, is a part of the cell synchronization, it was the cell size of the somatic cells when they are in the right cycle and the expression of certain RNA and certain sizes, which one. Then I was always looking for tiny, small cells that in theory, those were in the GOG1 of the cell cycle. I injected at the perivitelline space, making sure that the cell hopefully is in contact with the olema to avoid that during your electrofusion is not going to get stuck or stick here at the perivitelline, at the zona pellucida, then they don't fuse. Then it's coming the electrofusion, is basically to passing some electrical pulses to make this cell to fuse with the olema. I use something that we call it chopsticks. It's not the normal, most of the people use chambers. We use chopsticks, it's a Japanese equipment, high voltage. And basically these two membranes make pores, electroporate, and the somatic cells get inside the cytoplasm that we confirm like 30 minutes after. After the confirmation of that your couplet is reconstructed, we activate the embryo with several chemicals that we use, cycloheximide and electrical activation, but you can use several types of activation, inducing similar a calcium release inside and a lot of the growth transcription factors they are inside to helping the reprogramming of the cell and initiate the embryo. That you have to do with one and several of those during one day to maximize the possibility of obtaining good quality embryos. After that, okay, we did that, we did a lot of research and we produced kittens. But look what happened. The first 9 kittens born after all this process, embryo transfers, a lot of work, all these kittens die or were stillborn or they were or dying within 36 hours. We knew by then that by other research performed in different mammalian species about the abnormal reprogramming, epigenetic errors that happen, but how to correct those to produce live offspring. Then the main causes of death that we found was fetal absorption and abortions. The normal placental dysfunction reported is a lot, especially in bovine clone animals. The lung immortality and failure respiratory, that it was one of the strongest epigenetic events of abnormal reprogramming in our kittens. Physical abnormalities and septicemia. But how to overcome each of these factors to produce at least live, healthy clone kittens? Then we start with the placental abnormalities. We saw this is on the left, normal placenta, and we constantly see that the placentas have reduction in thickness and a lot of atrophy everywhere. But still, this placenta carry pregnancies to term. But what happened, what we could figure it out is that when the cat start labor, cats start to have contractions maybe even 5 days before labor, before real labor. And then when it's coming the real labor, this placenta have completely dissociated of the endometrium and the kittens die by hypoxia. Then how we treat these cats to avoid that these kittens die? We first start with progesterone. We administer progesterone that has benefits and negative things also, but we decide to administer progesterone first to maintain the corpus luteus and to try to inhibit as much as possible early contractions. Not always progesterone inhibited contractions in these cats. Then we move on to monitoring the contractions of the cat. We start to monitor these cats around day 55. And that was interesting because, of course, it's put the monitor in the belly of this domestic cat, and the son of the zookeepers always held and try that the cat stays still for 30 minutes twice or 3 times per day. After that, we send all the information to Colorado through telephone, and they tell us, okay, the contractions are strong enough that you need to inhibit the contractions. Then we start with the betamethasone by fixed dates, because we couldn't wait until the contractions start, because sometimes the betamethasone didn't inhibit contractions. By fixed dates, twice, if we were able to do it, and that helped us to reduce early delivery, early contractions, and also to program. We program when the kitten is ready for lung maturation, because we also mature the lungs. Another aspect that was really difficult to understand, it was septicemia. Some of the kittens just were born with septicemia, that they were immunosuppressed. They have a failure of passive transfer of maternal antibodies. We didn't know. Then we treat most of any clone kitten that was born immediately after born and during their first 24 hours of life. We administered blood cat serum subcutaneous to improve the IgGs in these animals, and antibiotic therapy. By modifying all these, we were able to produce a lot of healthy African wildcat clone kittens. And of course, we produce male and female healthy African wildcat clones. And we were happy about it, but it was a lot to learn. It was not only to produce live clones, it's produce healthy live clones, and how we can optimize all the technology that we were developing, but also understanding some basic science around this. One thing that was quite interesting with the clones that happened with them, it was this open abdominal wall with organ sterilization. We consult with several experts in cats, and some say that some species of cats that are quite inbred sometimes show this one. But it was, we never said in the institute, we never experienced this. Then we thought this is something that we are inducing with the cloning. What is going on? It has been reported as syndrome in babies, in human babies, which are associated with the deregulation of some genes, and it was caused by the in vitro culture. When the in vitro culture wasn't not as optimized like today. Then we were thinking, okay, there are abnormal epigenetic events that are inducing these abnormalities. Possibly yes, but how to overcome this? We know what happened during the reprogramming of the somatic cell nuclear transfer. We know that we have a somatic cell, a fibrillous cell, that basically is a fibrillous. I'm not an embryo. I have some of my genes are turned off, and I have a lot of methylation on those genes to avoid expression. As soon as we put that somatic cells with the oocyte, there are so many factors inside the oocyte that help and produce an embryo that moves, that alter the epigenetic events of those cells, and basically change for acetylation, hyperacetylase, some of the histones on that DNA, and activate the embryonic genes. That if occurs and everything is normal, but a lot of abnormalities going on here because sometimes those genes are not turning on. At that moment, we knew that, okay, we have these cells, what we can do with these cells to enhance the reprogramming. And it is known that the in vitro culture affect the somatic cells in different ways. We saw it that how the numbers of chromosomes, we can lose chromosomes during in vitro culture, how we need to, by then we were testing each cell line to know how many passages we can have the cell line in vitro to know that I have normal content of chromosomes. But however, we didn't know much about how the epigenetics events of those cells were affected during in vitro culture. Then our common practice by then was to using cryopreserved somatic cells because it was practical. It was easy to have those cells in culture already synchronizing the cell cycle stage that we want, freezing them, thawing them just before cloning and produce our embryos. However, it has been already reported that the development in some clone animals of the embryos to the blastocyst stage could be reduced when you use frozen and thawed cells, and also maybe increase fetal losses. Then we start to evaluate how the cryopreservation of the cells can affect these epigenetic events. I'm going to go quick about just a couple of things that we do. We evaluate our cells, we test our cells for the acetylation of the histone 3 lysin 9, that it has been shown that if it's acetylated, it helps to reprogram the cells better, it's turn the genes on better. But if there is methylated, this doesn't turn on this gene. Then we evaluate the acetylation level of those cells in 3 different cell lines. This is sand-cat cells. These are not African wildcat cells any longer. And then we clearly see these are exactly the same cell lines. When we tested immediately after thawing, maybe an hour after thawing, they were completely hypoacetylated. But if we culture the cells at least for 4 to 5 days, they become hyperacetylated. That means we demonstrated that the cryopreservation per se affect the acetylation. I am not putting any of the other data we have here, but we have demonstrated the same in clone, in somatic cells, in bovine somatic cells, and we demonstrate the same in vitrifying embryos, cat vitrifying embryos, how no more vitrification affect the acetylation and affect the epigenetic events of embryos. That means the culture of cells and embryos in vitro culture affect the epigenetic events of the cells. Then we evaluate the OX4 expression at RMA level of domestic cat embryos as a control, knowing that this is interspecies clone embryos, but it's still as like a control of how is the expression on OX4 on those embryos. And we saw that most of the embryos express OX4. And look, just embryos, cat embryos reconstructed with cells that were frozen and thawed and used immediately for cloning, just 25% of the embryos. That means they were abnormal reprogramming. And when we cultured the cells, we got similar expression of the OX4. That means we could demonstrate that really the changes were on the cells. Similarly, we saw it when we evaluate the OX4 at protein level, but with the difference that these were cat clone embryos that were cultured, they were produced with cells that were cultured, that we have embryos that express OX4, but the distribution of that OX4 was still abnormal. However, there were not a lot of embryos, and there is people saying that is detectable, the protein expression can be also detected in the trophodector. By doing that, we transfer embryos with cat embryos to produce cat clone kittens with cells that were frozen and thawed and immediately transferred and cultured. And look how interesting it is here. All the embryos that were producing with that type of cells were reabsorbed of abnormal kittens. With these 3 kittens had organ sterilization. While we're just by culturing, we reduce substantially the amount of embryo reabsorbed, and all kittens produced were morphologically normal. That is one of the big improvements of that. And we did a lot of work that I'm not presenting about epigenetic events in black-footed cat embryos. We produced 1 caracal clone kitten, and it's amazing the technology, how can be used appropriate to help the preservation of those species. And then by the end, it's showing that most of these clone animals were fertile. We bred one of the clones with 2 of the female clones, and we produced normal kitten. All those kittens were characterized by RNA, were characterized mitochondria, they were homoplasmic, heteroplasmic, and they were a mix of things. Most of them carry mitochondria from mothers, but a lot of them, they were also heteroplasmic. Okay, this is a job that, believe me, it was 18 years, extraordinary job. This is some of the people that work, but there are a lot of more people that were involved in all our projects, and especially Dr. L. Pop, that was the pioneer on all of these technologies. Okay, thank you. And now questions.
Thank you very much, Martha. That, of course, you're talking my language, so that was a wonderful talk. I really enjoyed the history and the span of topics that are involved in somatic cell nuclear transfer. We're going to leave the question and answers for the end, but I'm going to just ask you a quick question, something that I'm really interested in, which is that you focused a little bit on the donor cell cultures, of course, which is, some of the meat of cloning. So my question, are all fibroblasts created equally? What are your thoughts on the fact that we don't really have standardized criteria for assessing donor cells for cloning? And I put this in perspective that, all zoos have banked fibroblasts over the last, 30, 40, 50 years. Is all that material valuable, do you think?
I think that it is. The only, any material that you can preserve, I really believe in biobanking. I don't know how expensive biobanking can become. especially when you have tanks and tanks and tanks, and what the heck I'm doing here? Like you say, what I am doing here. I think that I can tell you, for example, with just the son of the species, I don't forget right now, one of the species that I work, the rusty spotted cat, for example, the female cell line work really well. 20-30% of blastocysts, good quality. The male somatic cell line, really bad. If I get 1%, I couldn't. One of my biggest effort were with black-footed cats. And I used like 4 different cell lines, and it was terrible. We get implant, and then I did a lot of work in epigenetic events in embryos, in cells, trying to understand the methylation of those cells, the methylation. And I work a lot with embryos, changing, modifying the epigenetic events on embryos and in cells. And interestingly, the best result always is focus on the cell. Don't bother with the embryo, because you are modifying it. But at the end, it's the cell that is going. And these cells behave, they were 4 different females and males, and behave the same. That means it's a huge difference between cell lines. If it's between a species, where you are going to put it, you name it.
So you hit the nail on the head, and you know, I'm happy that's where you took the answer, because those are bits of information I think everyone needs to know. So I'm going to pick your brain a little bit more at the end, but thank you for sharing that answer with me. And for now, we're going to move on to the next speaker in our series. So thank you again, Martha. We now move on to Dr. Richard Behringer. He joins us today from Houston, Texas, where he is a professor in the Department of Genetics and the Ben F. Love Chair for Cancer Research at the University of Texas's MD Anderson Cancer Center. Dr. Behringer's research focuses on mammalian developmental genetics, which includes organogenesis, stem cells, evolution, and you know, he's a busy man. He conducts field studies of marsupials on Kangaroo Island in Australia and bats on the Caribbean island of Trinidad. He's also been director of both the molecular embryology of the mouse course at Cold Spring Harbor Lab and embryology course at the Marine Biological Lab in Woods Hole. He has edited, he has published, and done a lot of other amazing things, but I'd like to finish by saying he is an advocate for developmental biology, genetics, and reproductive biology. So on to you, Professor Behringer.
Okay. Can you hear me okay?
Yes,
okay.
Thank you.
All right, well, this is really a special treat for me to be participating in such a global get-together of reproductive biologists. Today, I want to talk about a signaling pathway that regulates sex differentiation and how that pathway varies across vertebrate species. And well, it kind of all starts with a fundamental question that we have, and that is, what controls the differentiation of an embryo into a male or female? So most of our studies are in the mouse, but towards the end of the talk, you'll hear about the other animals that we are working on. So I'm going to show you the mouse reproductive organs, in case you're not familiar with the mouse. So these are the adult reproductive organs from a male and a female. And so in the male, just for orientation, this is the bladder. These are the seminal vesicles, the vas deferens coming into the epididymis, and this is the testis, and there's a testicular fat pad. So those are the male organs. And on this side are the female adult organs. The mouse has a bicornuate uterus, so these are uterine horns where multiple fetuses can implant and develop. You probably can't see it, but there's a very coiled tube here. That's the oviduct. And this pinkish area is the ovary, and there's an ovarian fat pad. So these are the adult mouse reproductive organs. And we just asked a simple question. So we have these sex-specific organs that form in males and females. How do embryos generate these organs? Okay, so a lot of my research starts with this guy, Johannes Müller. In 1830, he published a paper in German, which makes it a little hard for me, but I've been translating it recently. So the translation is Development of the Genitals, Anatomical Studies on Human and Animal Embryos. And he looked at the developing reproductive organs in many different types of vertebrates. So mammals, birds, reptiles, amphibians, and probably not the typical organisms that we study today. And what he found was a pair of ducts that are now known as the Müllerian ducts. And the fundamental discovery was that these ducts give rise to a uterus and oviducts depending on the species. So a lot of my research focuses on these Müllerian ducts. In the mouse, we are able to dissect out some fetal organs and place them in organ culture. And in this scenario, we take the urogenital system, and I think this was around a 12.5-day mouse embryo. Mice have a 20-day gestation. And then we place this fetal organ on a filter and then grow them in a medium air interface. And they'll continue to develop for at least 24 hours. And in this situation, we have labeled the Müllerian ducts with a yellow fluorescent protein transgene. So just to orient you, this is left and right sides. There's a pair of fetal gonads, which you cannot see, mesonephros, the fetal kidney, and embedded within the mesonephros is this Müllerian duct, an epithelial tube that starts up in the anterior region and it will progress to the posterior. So here's a time-lapse movie of the development of the Müllerian ducts. And so they're forming from an anterior to a posterior position, and ultimately they'll fuse with the urogenital sinus. So what's interesting is that, as I told you, the Müllerian ducts give rise to the uterus and oviducts. The interesting thing is that the Müllerian ducts form in both female and male embryos, but males do not have a uterus and oviducts. So there must be some type of mechanism to eliminate the Müllerian ducts in a male. Otherwise, you'll have a male that forms a uterus and oviducts. Okay, so this is a diagram of male sex development. And let's look what's going on. We have a pair of gonads. This is around 13.5 days in the mouse. So we have a pair of gonads. This gray area is the fetal kidney called the mesonephros. This blue line is an epithelial tube called the mesonephric or Wolffian duct. That forms first. And then the Müllerian duct here in red forms after the Wolffian duct. So at some point, these gonads will become a testis or an ovary. If it is a testis, it will produce testosterone, which will lead to the differentiation of the Wolffian duct into epididymis, vas deferens, seminal vesicles, so male-specific organs. The fetal testis also expresses anti-Müllerian hormone, or AMH, and that actively eliminates the Müllerian ducts in a male embryo so that males don't form a uterus and oviducts. So the Wolffian ducts differentiate into male organs, whereas the Müllerian ducts that would usually give rise to female organs are eliminated in male embryos. So what does that look like? This is a mouse embryo at 14.5 days. It's a male. So these are testes. And it's carrying a transgene that expresses beta-galactosidase and will stain blue. And it happens to mark the Müllerian duct and the Wolffian duct epithelium. So anti-Müllerian hormone, AMH, is secreted by the fetal testes here. And the hormone reaches the Mullerian duct and interacts with the mesenchymal cells surrounding the duct, not the epithelial tube itself. And the hormone AMH interacts with a receptor, which is called AMHR2. And that leads to the elimination of the Mullerian system. So I just showed you this image. The female has her Mullerian system formed. She also has the Wolffian duct still. This is one day later, and maybe you can see that the Wolffian duct, this lighter blue, is still there, but there are gaps in the Mullerian duct. So the Mullerian duct is actively being eliminated at this stage by AMH. Okay, so many years ago now, we used gene targeting and embryonic stem cell technologies to make knockouts in the gene for the hormone AMH and the gene for the receptor AMHR2. Interestingly, the phenotypes are identical. And the phenotype is predominantly in the male mutants. So these are adult reproductive tracts from a wild type male. And these are the organs I've shown you before. These are the testes, epididymis, vas deferens, and seminal vesicles. In this case, I'm only showing in the receptor mutant, but the AMH mutant would be identical. So these mutants look like males, behave like males, and they actually have all the male organs. So they have testes, epididymis, vas deferens, and seminal vesicles. But in the absence of the hormone or the receptor, they also have a uterus, which is shown here. And if you follow the uterus up here, there's also an oviduct. So these were experimentally generated, but in humans, there is a syndrome called persistent Mullerian duct syndrome, where about 45% so it's an autosomal recessive disorder, and about 45% of the individuals with this syndrome, males, have mutations in the hormone gene AMH, 40% in the receptor AMHR2, and then the remainder is still unknown. And there is a uterus in each condition, and sometimes there is cryptorchidism, and it could be bilateral, unilateral, or both testes descended into one scrotum. Okay, what I'm showing you here is a diagram of the intron-exon structures, well, the exons of the AMH locus. There's 5 exons. There's also an AMHY locus in some fish. There's 7 exons. And for these genes, the actual active ligand coding region is the final exon. And this is the exons of AMHR2. And the pictures of the animals show the various species where there are mutations in these regions, in these genes. So I've told you about the mouse for the receptor and the ligand that we experimentally generated. And I told you about the humans, which I don't show here. There's also a dog model where there's an AMHR2 mutation, and those dogs are males with a uterus. The only other type of vertebrate where AMH or AMHR2 mutations have been generated are in bony fishes or teleos. And so what are the phenotypes of AMH or AMHR2 in fish? So it's very interesting, and it's very different from the mammalian mutants. So there are 2 phenotypes that I'll tell you about. One is exemplified here in the Medaka fish. Medaka has an XX-XY sex determination system. And I'm showing you a wild type and an AMHR2 mutant genetic females. So this is a dissection showing the ovary size in a control. And this is the receptor mutant showing this very, very big ovary. And the reason why it's so big is because there's uncontrolled germ cell proliferation. So an ovarian germ cell tumor, if you will. So that's one phenotype. The second phenotype is exemplified here with Nile tilapia, and they also have an XXXY system. And so this is the control genetic male XY, showing the seminiferous tubules and the active spermatogenesis. In an AMH mutant that's got the gene on the Y chromosome, a knockout, this gonad is an ovary, and you can see eggs being formed in this ovary. So the second phenotype in the bony fishes is male to female sex reversal and complete sex reversal. So I've told you about the AMH and AMHR2 mutations in mammals and bony fishes. So let's compare them. Mammals have Mullerian ducts. Actually, bony fishes don't have a Mullerian duct, yet they still have these genes. In mammals, there are humans and dogs that have spontaneous mutations in these genes. We engineered them in mouse, and they have a common phenotype, and that's a male with a uterus. These are the various bony fish species in which these genes have been manipulated. And there's the two phenotypes. One is that males become females, so sex reversal, and mutant females have excessive germ cell proliferation. So we have these common genes that they're conserved. They have phenotypes and reproductive organs, but the phenotypes are quite different. And so we started to wonder, what is the function of AMH in other vertebrate species? This is showing a vertebrate phylogeny, and I've told you about the mutant phenotypes for AMH and AMHR2 in mammals and the bony fishes. And when you look at the evolutionary relationship between mammals and bony fishes, you'll notice that the amphibians would be an interesting type of animal to examine. So we've done that. We are collaborating with the National Xenopus Resource at the Marine Biological Laboratory in Woods Hole. And we're working with the director, Marco Horb, and his scientist, Danielle Jordan. And we're working with a specific species of frogs, and this is Xenopus tropicalis, for a number of reasons. One, it's diploid, whereas Xenopus laevis, which is much more common, is allotetraploid, which would make genetics quite difficult or challenging. The other thing is that Xenopus laevis has a very long cycle to make the next generation, whereas Xenopus tropicalis is much quicker. It takes about 6 to 8 months to get the next generation. So we're making the mutation in the AMH and AMHR2 genes in the tropicalis frogs. So we've designed CRISPR guides, RNAs here for the AMH exon 2, which is right here. The ligand encoding region is here in exon 5. So we were thinking that if we block expression here, we should create a knockout. So we injected the CRISPR components into frog eggs, let those grow up to be frogs, and then genotype the founders. And we've isolated a number of alleles at the locus. And I'll just show you this one allele. It's a 5 base pair deletion right around here. And that throws the reading frame out of frame and then a premature termination. So these mutants should not make functional AMH. So we had our founders that we isolated. We then bred the founders to generate F1s, heterozygotes, waited till they're sexually mature, and then bred them together to generate the F2 animals. And so this is where we're at. We have AMH mutant frogs. We genotype the frogs by taking a web punch. And in the first 14 animals that we've genotyped, we have 4 wild types, 7 heterozygous, and 3 homozygous mutants. So the homozygous mutants are viable, and we're obtaining them in the predicted Mendelian ratios. Xenopus tropicalis does not have sex chromosomes, but a single nucleotide polymorphism has been identified that can be used to genotype the sex. So we're currently genotyping the frogs to see what phenotypes they might have. Okay, well, those are frogs. We've turned our attention to reptiles. In 2019, Doug Menke's group at the University of Georgia created the first genetically modified reptiles ever, and they use CRISPR. The approach was quite remarkable. They take a female lizard, open up the abdomen, visualize the ovary, and then they directly microinject follicles with the CRISPR components. They put the ovary back in, close up the wound. These are wild-caught lizards, so they're actually already impregnated with stored sperm. And then they wait for the lizards to lay eggs. And if you're following, they're manipulating the oocyte. So then if this has been bred with a wild-type male, you should get heterozygotes. The remarkable thing, so the gene they tested this on was encodes an enzyme called tyrosinase. And if you don't have tyrosinase, you have no pigmentation. And so albinism is caused by mutations in tyrosinase. And so this is a remarkable picture of lizards that have hatched out of the eggs from those manipulated females. Heterozygosity for a tyrosinase mutation should be wild type in pigmentation, yet they have an albino. The speculation is that the injection of the CRISPR components directly into an oocyte in vivo not only edited the oocyte chromosome, but when the wild type sperm came in to fertilize, the CRISPR components were still there and acted on the sperm DNA to then create this homozygous mutant. The exciting thing is that in one manipulation, you can generate a homozygous mutant in one generation. So what are we up to? We have created guide RNAs for the AMH locus in these anole lizards, and we should be injecting those later this summer. And then finally, there are new methods to modify the germline of chickens. And it's very interesting. In a chick embryo at this stage, the primordial germ cells are in the blood circulating. So you can take the blood, culture the cells, and primordial germ cells will persist and proliferate. Those can be modified transgenically or with CRISPR. And then once you have the modified cells, you can just inject them back into the circulation of an embryo and then have chicks born with the manipulation. And so basically you're making germline replace chicken chimeras. And so we've been able to successfully isolate primordial germ cell cultures from the chicken blood, both male and female, and we're in the process of genetically modifying them to mutate the AMH locus and the AMHR2. So I've told you that there are mutations in this pathway in mammals and bony fishes. We've now made the mutation in amphibians. We're headed towards making the mutations in lizards and birds. And I'm thinking this may be one of the first broad cross-species studies on a signaling pathway to examine over evolution, how has that been modified to regulate different aspects of reproductive biology and sex development. So these are the people who've contributed to these studies. And considering my audience, I just want to make a pitch. We have a cryozoo of frozen fibroblasts from nearly 300 mammalian species that was initiated by Dr. T.C. Su. If you're interested, please contact me, and I'll stop there and thank you.
Thank you very much, Richard, for this beautiful overview on very basic developmental biology. And you're right, you cover very, I was taking notes as you were speaking, saying, oh yeah, I'm going to ask a question about, and what about that species? And what about the species? And you were coming up with, so this is great. So in the interest of time, I think we will keep questions for the final Q&A session. But
Okay.
we're gonna thank you so much again, Richard. And I would like to introduce now our third and last speaker, Dr. Hiroshi Kiyonari, who is a scientist from Japan. He has obtained his PhD from Kumamoto University, and then he became a team leader at the Laboratory for Animal Resources and Genetic Engineering at the Riken Institute in Kobe, Japan. He specialized in reproductive biology and embryology, but importantly enough, he's also really, besides managing really this center, he's also producing a lot of genetically engineered mice models every year in collaboration with a lot of biomedical research institutes in Japan, but as well as in Asia. So welcome, Dr. Hiroshi Kiyonari.
My name is Hiroshi Kiyonari, Riken BDR Japan. I'm very happy to be able to talk to you today. I'm Animal Facility Manager in Riken BDR, and I support BDR investigators in various aspects of laboratory animal experiments. In addition, my laboratory has aimed to develop new genetic and developmental biology tools in the mouse and the other small vertebrate species. So today, I'd like to talk about the transgenic technology in new animal model we recently developed. I have no financial relationships to disclose. The mouse is the most widely used animal species in studies of mammalian genetics, developmental biology, human diseases, and so on. There are many reasons for this, but the most important one, I think, is that the technology for producing genetically engineered mice has been established. Mice have been established the position as model animals through the development of transgenic mice by random transgenesis, followed by the development of knockout and knock-in mice by the gene targeting using ES cells by Dr. Evans, Capetti, and Smiths, who won the Nobel Prize in 2007. However, the production of knockout and knock-in animals by gene targeting has been limited to very few animal species with established ES cells, such as mice. In the 2000s, the genome editing technologies has accelerated. In 2013, genome-edited mice were generated using the CRISPR-Cas9 system, which is the immunosystem of bacteria and archaebacteria. Co-injection of Cas9 encoding messenger RNA and target sequence single-guide RNA into mouse zygote resulted in generation of not only monoallelic mutants, but also biallelic mutants with high efficiency in one generation, of course, without ES cells. With the development of genome editing technology, it has become possible to generate genetically engineered animals, even in animal species in which ES cells have not been established. I think this is the most innovative technology of the genome editing. Among vertebrates, genetically engineered animals have not been generated for monotaurine and marsupial. The development of a monotaurine laboratory animal model is difficult because there is only a single egg laying per year, and very few eggs are laid each time. In contrast, some marsupial animals may have potential as new models for animal experiments. Marsupial have very unique characteristics not shared by other mammals. In particular, as you know, much of the development of premature babies born after a relatively short gestation takes place in the external environment. This is a phylogenetic tree of marsupials derived from retroposon data in previously reported. As you can see, the Monodelphis domestica, commonly known as a gray short-tailed opossum, plays as the first branch of the marsupial tree. The gray short-tailed opossum, hereinafter called the opossum, is one of very few established laboratory models in marsupials. Due to many biologically unique characteristics and experimentally advantageous features, the opossum is used as a prototype species for basic research on marsupial biology. For example, the opossum is similar to the mouse and rat in body size and breeding characteristics. In addition, like other marsupials, the opossum has a short gestation. After only 40 days of gestation, at birth, they are corresponding to embryonic day 13 to 15 in the mouse and rat, or around 40 days in human. Female opossum don't have a pouch. The baby clings to the mother's nipples until weaning. So we can continuously observe and analyze various developmental events, including organogenesis, using the same baby. And the opossums are the superior model animal of the melanoma. Other than humans, the opossum is the only naturally existing mammals which developed melanoma in response to ultraviolet radiation alone. In addition to these features, opossum provides a variable marsupial model for studying many other biological attributes. But despite such desirable features, in vivo studies of gene function in the opossum lag far behind those of eutherian mammals due to the lack of established methodologies for in vivo genetic manipulation. In order to tackle this technical hurdle, we aimed to develop a reliable platform for reproductive biology and zygote micromanipulation in this species. So today's topic is these three. To correct zygote, cooperating conformation after pairing is required. So we installed a custom video recording system to detect cooperation. Because unlike mice and rats, vaginal plug and detection of spermatozoa in vaginal smears are not reliable in opossums. The graphs show cooperation incidences during an observation period of 8 nights. Day 0 shows pairing day. All cooperation events were confirmed only between day 4 and day 7. Opossum is an induced ovulator and is estrused by such as male pheromone, but it takes time from pairing to mating due to its strong territorial. Such a wide window of cooperation timing is not desirable for the development of efficient production colonies suitable for zygote genetic manipulation in the opossum. So to narrow the time frame of cooperation, we tested whether hormone stimulation can promote cooperation by estrus induction. high dose of PMSG 30 IU and 5 IU made it possible to shorten the window of copulation timing. But neither of these conditions resulted in the acquisition of zygotes. On the other hand, low dose of PMSG induced copulation throughout the observation period. The majority copulated by day 3, and we could get zygote from them. In addition, to limit the cooperating data a little more, we injected HCG 72 hours after PMSG. Additional injection of 1 IU HCG resulted in more than 70% of females cooperating within 4 nights of pairing. Furthermore, more than 50% of females injected with a higher dose of HCG cooperated on the day or the next day of pairing. And we could also obtain zygotes from them. These results indicated that the hormone stimulation can induce copulation within a short time window than natural mating. In particular, injection of 1 IU PMSG and 5 IU HCG combination was made possible to drastically narrow the overall range of copulation timing. The next important task was preparing pseudo-pregnant females for embryo transfer. In rodents, the estrous cycle is easily identified by the appearance of the vulva or by measuring impedance in the vagina. Embryo transfer is typically performed in pseudo-pregnant females prepared by copulation with vasectomized males. However, in the case of the opossum, there has been no effective method established to obtain timed pseudo-pregnant females. To alleviate this technical issue, we attempted to produce pseudo-pregnant recipients by letting females cooperate with vasectomized males upon estrus induction by only pairing or hormone administration. Cooperation was confirmed by video recording, and pseudo-pregnancy was further verified by uterine swelling at the time of embryo transfer. As you can see, both pseudo-pregnant female uterus are swelling as a pregnant female. About a third of untreated females cooperated between day 2 and day 7, whereas 27.4% of hormone-injected females cooperated during the first 3 nights. The cooperation rate of females for pseudo-pregnancy appeared lower compared to those for zygote collection. This result is consistent with previous reports showing that vasectomized males were generally less likely to induce estrus in females than in intact males. Next, freshly collected zygotes were transferred into the uterus of pseudo-pregnant females. No pups were obtained from 2 females, which were hormone-induced, but 7 pups were obtained from the other females whose estrus was pairing-induced. all of them reached weaning in healthy conditions. These results indicated that induction of estrus and subsequent copulation were successful to produce a pseudo-pregnant recipient by pairing with a vasectomized male. We succeeded the establishment of a scheme for zygote collection and embryo transfer. So to explore the possibility of genome editing in the opossum, we attempted to target the tyrosinase gene with the CRISPR-Cas9 system in pronuclear stage zygotes by microinjection. The tyrosinase gene has been commonly used as a target to test the efficiency of CRISPR-Cas9 mediated genome editing in various organisms, because the resulting phenotype of pigmentation loss is easy to assess. Here is an example of a lizard, salmon, and rabbit. The opossum tyrosinase gene on chromosome 4 consists of 5 exons, and 2 guide RNAs were designed to target exon 4, which corresponds to exon 4 in the mouse previously used for highly efficient tyrosinase gene knockout. Opossum pronuclear stage zygotes were coated with thick mucoid layer and hard shell layer. So as shown in this movie, it was difficult to penetrate opossum zygote under our conventional microinjection setting for mice and rats. To overcome this, we used a piezoelectric actuator which can improve the penetration of injection needles into zygotes. Piezo is often used in intracytoplasmic sperm injection and nuclear transfer experiments. In addition, last year, we reported that piezo-assisted pronuclear injection significantly improves survival rate in the injected mouse embryos. This movie is piezo-assisted pronuclear microinjection for opossum zygotes. As you can see, the injection needle can be smoothly penetrated into zygote. We co-injected the guide RNAs and Cas9 protein into the pronucleus of zygote or into the cytoplasm when pronuclei couldn't be clearly identified. Injected zygote were transferred into the uterus of pseudo-pregnant female copulated with vasectomized male. The result of injection are shown here. 131 out of 133 injected zygote were transferred into 8 pseudo-pregnant females, and 6 females gave birth to a total of 19 offspring. Among them, 7 offspring exhibited readily detectable changes in coat color, ranging from complete white to mosaic, loss of pigmentation. PCR and sequence analysis on the target sites confirmed that nearly 80% offspring showed evidence of indel alleles. In total, we obtained 5 albinos, 2 coat color mosaics, and 8 with wild-type coat color carrying genome-edited alleles from all recipients which gave birth. Subsequently, a pair of mosaic female and albino male were crossed to confirm germline transmission. All 9 F1 offspring were albinos, indicated that both mutant alleles have been passed on onto the next generation. These results clearly demonstrated successful generation of tyrosines gene knockout of opossums by CRISPR-Cas9 mediated genome editing and germline transmission of genome-edited alleles to the next generation. We succeeded generation of genetically engineered opossum through the new technologies we developed in this study. This is the first report for generating of genetically engineered animal in mouse cells. It will contribute to the acceleration of in vivo studies of gene function in the mouse cells, which significantly behind those of eutherian mammals. Our current study provides a fundamental platform towards the establishment of the opossum as a unique and accessible genetic model system to explore comparative in vivo gene function studies in mammals. This study was carried out with the cooperation of these people. In particular, Mari, Aki, and Rico maintained and bred opossums, including the confirmation of copulation by video, and also contributed zygote correction and transfer into uterus. Kenichi is a microinjection specialist. All microinjection experiments were performed by him. And Takaya was responsible for preparing a reagent for microinjection and analyzing the offspring. And this study supported by JSPS Kakenhi. Thank you for your attention. Thank you very much.
Thank you, Dr. Kiyonari. That was a wonderful talk, as they all were today. And there you are in the flesh. Thank you for joining us in the strange hours of the day, wherever you are. We're about to start the question and answers, but what I just wanted to wrap up your session by saying that, you're talking about this high tech, genome editing. And I think what we've forgotten is the amazing achievement, not only of working with embryos in a species that is not the mouse or the human or the dog, but that you were able to do embryo transfers and the live births. So I congratulate you on that achievement. Those of us that work with wildlife are amazed today. So Pierre is going to help us start the question and answer period. And then hopefully let's bring the three speakers online. Let's have your cameras on and we'll start some of the questions for you.
Thank you, Gabriela. Yes, and I would like to join Gabriela in congratulating Dr. Kiyonari also for this huge achievement. Yeah, congratulations. So let's start with some of the questions that we received from the Q&A, and some of those questions were already answered during the presentations, but we kept some of the questions that we would like to be answered live. So I'm going to go back to Dr. Richard Behringer, and there is a question actually that is for the whole audience, because so it's a question from Jaswant Singh, who wants to know, who says that of course AMH plays a key role in ovarian reserve in mammals. It's used as an indicator. But Richard, can you comment on the role of AMH system in the adult females in other vertebrate classes for the same kind of evaluation?
There's not a lot of information. There's one study in the mouse knockouts for AMH, and those females are fertile, and actually they had pretty good litter size, but what was found is that they used up their ovarian reserve much quicker than controls. So the conclusion of that study was that AMH was regulating follicle recruitment. That's the only study on AMH signaling mutants that I know about in females. I am not aware of any human homozygotes for AMH or AMHR2 that have been described. In the fish, it's the germ cell tumors, the overproliferation. Other than that, there's no information on any other species. And I do want to say congratulations to Hiroshi. That was a stunning achievement.
Thank you.
Okay, so I have a second question for you, Richard, and then we will switch speaker. The question I have is also related to environment, because so you described really across different species, the role of this AMH system. But are you aware of any environmental disruptors in nature that can also affect really this AMH system and then have a detrimental effect on, of course, on the reproduction of wild species?
In the mouse studies that we did have one transgenic where we overexpressed AMH, and some of the males were normal looking and fertile, but we had some that superficially looked like females. Although when you open them up, they had very small undescended testes. They did not have a Mullerian system, and the Wolffian system was very underdeveloped, suggesting that by overexpressing AMH, the androgen signaling was somehow altered. Is there an environmental connection, exposures or something like that, that would alter AMH signaling? I guess it's possible. I'm very intrigued to make the mutants in the other species, because a lot of those other species, for instance, steroid hormones can influence sex development quite significantly, whereas in the eutherian mammal, not so much. So I think it's still awaiting more mutations in other species.
Thank you very much, Richard. Go ahead, Gabriela.
I'm going to jump back before we get to Martha again. I'm going to jump to Hiroshi a little bit. The first question is fairly technical. When you use, if in vivo matured oocytes are used as your recipient cytoplasm, do you see higher cloning efficiency than in vitro matured? So it comes back to that question of in vitro versus in vivo matured oocytes. Is one better than the other for cloning?
For me?
So this one's to Hiroshi, but from one of our participants.
But he wasn't cloning.
Okay, so it was put, that's, I wasn't sure if we were getting ourselves all confused. So Martha, please go ahead and jump in vivo versus in vitro matured.
Yeah, you will see difference, huge difference. That doesn't mean that you cannot have clone embryos from in vitro mature and produce live offspring. But the quality, especially of the cytoplasmic maturation, is what you are looking at, is much better in vivo. Optimization of the in vitro culture or in vitro maturation may help to have better cytoplasmic maturation, because people always talk about DNA maturation, but not about the cytoplasmic maturation. It's possible to use, but of course, it's much better in vivo, all sides.
And it's kind of a decade-long story, or a 2-decade-long story that we've all worked on. So to be a little bit more specific for you, Hiroshi, again, fairly, basic technical question for you, but, you showed the chart with all the different taxa where gene editing had been attempted. And those of us that are comparative reproductive physiologists and trying to work with too many species, is the gene editing technique equally efficient? Is this an easily translatable technique across taxa? Moving from mammals to lizards to birds, is it easily possible to move across?
Yes, I think, but some species is a little bit difficult because it's need to prepare the pseudo pregnant female. So sometimes it's very difficult to prepare them. So it's not easy, but it's possible, I think.
Okay. And of course, we're talking about the technical parts of it, as you know, Martha with the cloning, you and gene editing and the genomics that Richard talked about. But behind a lot of the work, we can't forget that then embryo transfers and in vitro embryo development has to happen in species where we may not have a textbook on it. So coming back to Richard quickly, then one of the questions from the participants is, have you tried CRISPR using stem cells?
We haven't. We have a extensive experience with mouse embryonic stem cells, human embryonic stem cells. We dabbled with rat, but those were in the old gene targeting days, pre-CRISPR. So we have not applied CRISPR to stem cells. We've been going directly into zygotes, and when we go to the lizards, we'll go directly into ovarian follicles.
Okay, very interesting. Thank you for that.
Yeah. In some cases, you may want to use the CRISPR technology in stem cells if you have a specific purpose.
Very good. Thank you. So jumping from there to Martha again. Sorry, I'm moving around here. So you had talked quite a bit about some of the imprinting problems, some of the challenges with the cloning technique. And one of the questions for you was, are there still, imprinting problems, aging problems, some of the things that they had been talking about for years from Dolly onwards?
Okay. There is interesting question because when you have apparently normal animals, they look normal and behave normal all the life. We have that with the African wild cats, with domestic cats. But at the end, we don't know if there are some epigenetic events that these animals are different, but could be normal, but modifications on the epigenetics that apparently they behave normal, but we change something. In theory or in practical, in some point of view, when an animal survives and is healthy, it won't show anything later. It shows early in the development. That means it's easy to know. For example, we have one of the African wild cats that have digestive problems, and is abnormal reprogramming. And he was relatively healthy all his life with that issue that you have to intervene all the time and treat it, but you don't want those side effects, but that can happen.
Again, interesting to get insight from those of you doing this every day, shall we say. I'm going to jump to Hiroshi now. So perhaps tell us a little bit about the advantages of creating these models again, just to kind of refresh our understanding for the expense and the time and energy of creating these models.
Yeah, so as I mentioned before, so in the possum, some organogenesis occurs after birth, and organs matured before birth will develop in a very short period compared with other mammals. So I think that it may be a big different gene expression pattern and the timing in organogenesis between Marsupial and Eucelians. So I'm interested in exploring key genes for organ development. So exploring the key genes for mouse pairs organogenesis using genetically engineered opossum will lead to an understanding of the mechanisms of mammalian organogenesis. And furthermore, it may be useful for regenerative study using iPS or ES cells, such as organoid study. Yeah.
Very good. And thank you for wrapping that up for us. Do any of our speakers have anything they would like to add related to their talks today to the participants online? We're coming to the end of our question and answers. So if there's any comments you'd like to say, otherwise I will bring, yes, Richard, go ahead, please.
I have a question for Hiroshi. Can you freeze the oocyte zygote sperm in the monodelphis?
I don't know. I was trying the fleeting embryos, but with same techniques in the mouse.
Yeah.
But it's difficult. It's because they have a shell layer. So some freeze reagent cannot into the inside of embryos. So so we have to remove the hard shell layer, but shell layer needs to develop the early embryos.
Yeah.
So it's
challenging.
Yes.
So on that note, I would like to thank everybody for taking the time to participate in today's session and the discussion. I would particularly like to thank the I3 speakers, Martha Gomez, Richard Behringer, and Hiroshi Kiyonari, and of course my co-host Pierre Comizzoli. Thank you as well to the organizing committee of the initiative, Jacques Cohen, Peter Nagy, Giles Palmer, Thomas Elliott, Shesta Sadruddin, Marianne Cevetes, and today's everyone who's working to help me behind the scenes with the question and answers, of course the participants as well. This session and the presentations will be available on the website soon. Don't forget to rate our lectures. It's very easy. And your certificate of attendance will be sent before the commencement of our next session, which is on Tuesdays, as always, at 3 p.m. Eastern Standard Time, 8 p.m. UK time, and 9 p.m. Central European time. So thank you, speakers. Thank you, participants. I definitely learned a lot today. Thank you very much. And bye for now.